Hello to everyone online. On behalf of the Northwest Territories Council on Invasive Species, Pests, and Pathogens, I'd like to thank you all for joining us today for the third in our series of webinars to celebrate National Invasive Species Action Week. Today, we are lucky to have a talk by Cory Olson on bats, bridges, and tracking the invasive fungus that causes white nose syndrome. Cory Olson is a professional biologist and coordinator of Wildlife Conservation Society Canada's Alberta Bat Program. He has worked with bats for 15 years, which began with his graduate research at the University of Calgary, focusing on bats of the boreal forest. He has conducted surveys of bats across Western Canada and has prepared several technical reports relating to bat management and conservation in Canada. He is leading the largest scale study to date of how bats in Canada use bridges. And with that, I'll pass things over to Cory to tell us more about that. Over to you, Cory.
Yeah, thank you. Thanks everyone for joining me for this webinar. I'm going to be discussing white nose syndrome monitoring in Western Canada. And in particular, I'm going to give a quick overview of our project examining the use of bridges by bats. This project is run by Wildlife Conservation Society Canada as part of the Western Bat Program. If anybody wants to follow our project, we have quite a bit of information on our website, albertabats.ca. We have various social media accounts where we post regular updates, and we also have a website, albertabats.ca/bridges. I'm going to give a quick overview of white nose syndrome. As the name suggests, this disease is characterized by a white fungal growth on the muzzle of bats, as well as various other parts of the bat's skin membranes along the wing and ears. This disease was first documented around 2006, and in regions where this disease occurs, they've documented over 90% population declines. Just a couple examples, Graphite Mine in New York once had almost 200,000 bats, and it declined more than 99% to just over 2,000 bats. This is pretty typical across other bat hibernation sites. There's also one cave more recently in Montana that had almost 2,000 bats, and now it has nearly no bats at all. So in regions where this invasive fungus occurs, it has had devastating impacts on bat populations. Currently, over 10 million bats are estimated to have been killed. It's probably well over that number by now, and over 90% declines have been documented in affected species. Not all bats are affected, but a lot of the ones that hibernate in Canada over the winter are impacted by this disease. This fungus grows on bats while they hibernate. It doesn't grow on them at all during the summer. But when they hibernate, this fungus invades their skin membranes. It causes frequent arousals from hibernation. And as a result of that, they deplete their fat stores and simply starve to death. Keep in mind that it's about 6 months of the year that bats need to survive without food, which is quite a long time to go, and they really depend on those fat stores for survival. So anything that affects their energy budgets is potentially going to be a major risk to bats. As most of you are aware, the little brown myotis and northern myotis and the tricolored bat, which doesn't occur in Western Canada, are listed as endangered under the Federal Species at Risk Act. In Alberta, these are also listed as endangered under the Provincial Wildlife Act, and I believe in the Northwest Territories, these are listed as special concern. The provincial designation is more important than the federal one, given that most of the land is on land under the provincial jurisdiction. White nose syndrome is caused by an invasive fungus called Pseudogymnoascus destructans. That name is somewhat difficult to pronounce, so we tend to just refer to it as PD. Pseudogymnoascus is a genus that does occur naturally in North America as well as across the world. It tends to do best in cool climates. I was reading a paper recently that they isolated four different species of Pseudogymnoascus in Antarctica and sea sponges. So the genus itself is fairly widely distributed. This particular species, though, PD, does not occur naturally in North America. It's an invasive species that we think was brought from Europe or in the general vicinity of Europe. Pseudogymnoascus grows naturally in soil. It can some of the varieties feed on plant matter, and importantly, it can persist in cave soil even when bats are no longer present. So losing bats is not necessarily going to cause this fungus to become less common in their environment. So this is just a SEM image. Actually, I should go back to the other one. The image on the left is an SEM, a scanning electron microscope image of this fungus. It's a close-up view of the bat's nose from a published paper. And as you zoom in, you can see the characteristic fungal hyphae and conidia invading the bat's skin. It grows through the skin, digesting their skin membrane, causing irritation and lesions on the bat skin. There's just another view that's color-coded. Bats that have these infections have damage to their skin membrane. Keep in mind their wings. The skin that forms the wings is very, very thin, so even minor injuries can actually be quite detrimental to the bats. So bats that have this disease have lots of scar tissue. It can heal when they emerge from hibernation in the spring, but it leaves behind this scar tissue. We don't know how this fungus got to North America, but we do know from various observations that bats can hitch rides on cargo ships. There's lots of there are lots of ships that cross the Atlantic, and it's not uncommon for a bat to just crawl into a crevice and roost there, and then get transported across the ocean. So how it got here, we don't know, but there's certainly a lot of potential routes that it could have taken. It could also be from a recreational caver that visits a cave in Europe and then travels to North America, or maybe even a biologist. Fungi do quite well in cave environments. It's very humid. It's close to 100% humidity. Those are perfect environments for fungal growth. So when we visit cave hibernacula in North America, we quite often see fungal growth, although most of that is not from the PD fungus. This is a graph in the literature. It's showing the growth curve of this fungus at different temperatures. So this is a fungus that grows very well in cool environments. So the optimal growth is from about 12 to 16 degrees Celsius. That's on the cool side for bats during the summer. Most bats would be roosting in much warmer temperatures during the summer. But when they hibernate at about 7 degrees Celsius or so in the regions where this fungus has been around, which is well within the range where this fungus is going to grow quite well. Above about 20 degrees Celsius, the fungus doesn't grow, and below zero, the fungus doesn't grow. And one important distinction that I would emphasize is that as we go farther north in North America, the hibernating temperature has potential to decrease. So Here is one cave in the Canadian Rocky Mountains. It's in Alberta, in the foothills. And there's about 100 bats hibernating in this cave, but the conditions under which they're hibernating is just barely above zero. So it's about 1.6 degrees Celsius, um, which is really at the lower end of that growth curve. So it remains to be seen how our bats will be impacted by white nose syndrome, especially in more northern regions. The hibernating conditions do vary by region, but it's not a guarantee that bats in more northern regions will have the same catastrophic die-offs as in other regions. So what we really need is monitoring to better understand the impact that this disease will have on bats. This is the current spread of a PD in Canada, or I should say, yeah, so the image on the right is the current spread of PD in Canada. The image on the left is the spread or the confirmed detections of white nose syndrome. PD refers to the fungus that causes white nose syndrome. White nose syndrome refers to the actual disease. In Europe, they have the fungus PD, but they do not have white nose syndrome. So the distinction between PD and white nose syndrome is important for understanding how it's impacting bats. But before we started our project in 2021, the farthest east or the farthest west in Canada that we knew PD occurred was in Manitoba. There's lots of karst topography in Manitoba, and there's lots of caves that we know where bats hibernate. The same was not true, however, in Saskatchewan, Alberta, and British Columbia. We know of a few caves in Alberta and British Columbia where bats hibernate, but the number is very few. It accounts for barely any of the population. So for the most part, we don't know where bats in Western Canada are hibernating. However, for monitoring diseases like PD and white nose, or for monitoring diseases like white nose syndrome, we can rely on observations at summer roosting habitats. Our program began a community science program in 2016, where we were encouraging people to submit observations of bat roosts so that we can understand how bats are using anthropogenic habitats, but also use these as potential sites for supporting research projects and monitoring projects. This is a very typical building used by these bats. This is an old granary. And if you look up at the roof line of this building, you'd see a whole bunch of bats staring down at you, about 200 in this one building. And we get lots of such reports from buildings and bat houses especially. But you'll notice on this map that most of those observations are really clustered around population centers. This is Alberta. Most of those points are between just north of Edmonton and just south of Calgary. So for the purposes of disease monitoring, this isn't really ideal because we don't get a lot of spatial distribution in our sampling sites. One other human feature that has not received a lot of attention in Canada are these bridges. And we have some anecdotal observations from Alberta and Saskatchewan and British Columbia and elsewhere of bats using bridges as roosting sites, but nobody really understood how common using bridges was for bats. So there comes in our Western Canada Bats and Bridges project. The benefit of bridges is that even when there are not people around, there tends to still be bridges. So you can find bridges crossing rivers and creeks all the way up into the far north of Canada, albeit at lower densities as you go north. But there are still lots of sampling sites. So these are all bridges in Alberta, Saskatchewan, and Northwest Territories. Should also add that we're adding Yukon this year, and British Columbia has a pretty active survey of bridges going on currently as well. Our project, though, spans over 3,000 kilometers from the far northwest of of our project site all the way down to the far southeast. So this is a massive area. And not only are we sampling for disease in this area, but we're also collecting samples that can support DNA barcoding to determine bat species. So this is effectively the largest scale bat inventory that is currently being done in Western Canada. Before I go any further, though, I do want to emphasize that this is not just our organization contributing to this project. We've been spearheading it, but the project is only made possible through many partner organizations and government agencies and volunteers. And there are a lot of people contributing to this project that I want to recognize. Our survey of bridges consists of a visual inspection of these bridges. Using a flashlight, we can scan the various crevices of these bridges to look for bats. So there are some of our participants examining these bridges. And when we're looking for bats, there are a number of different ways that bats could be using these bridges. And the one that is perhaps the most of interest are these maternity colonies. Maternity colonies are groups of female bats, mothers, that are raising their pups. They only give birth to one pup per year for the most part, but they can live in groups that could number up to several hundred, sometimes even into the thousands of individuals. So they are not difficult to find, but bats hiding during the day tend to be really well hidden within deep crevices. This is a very typical bridge that we found bats in. It's not very common in the Northwest Territories, but across our study region, this is perhaps the most common bridge where we find these maternity colonies. And these beams, they fit together, but they leave a little bit of a gap, which are perfect for bats to hide in, kind of reminiscent of bat houses where they crawl into very narrow chambers. These bridges work very similar to that, and they can support hundreds of individuals. So this is just one section of the beam. If you scan your light way up at the top, you can see all these bats staring down at you. There's just a closer up view. And sometimes they're not maternity colonies, but instead are just used by these solitary bats. And this one is a big brown bat. These last bats I was showing were little brown bats. Big brown bats and little brown bats are the most common bats we see during the day. But bats also use bridges at night as night roosts. And we think that most of the bridges are being used as night roosts and not as maternity roosts. And the benefit of bridges is that they tend to be made of concrete, and concrete has a really high thermal mass. So it absorbs heat during the day, and then it releases it at night. So these are very warm structures for bats to roost on, and they can save energy by not having to heat their bodies quite as much. And it could be a huge benefit to these bats. But importantly, even the bats that are not necessarily using the bridges during the day might still be using them during the night. So we think that sampling at bridges could be a way of detecting species that are not commonly found using human structures during the day. So while we're looking for bats, what we're really trying to find are fecal deposits, and bat poop is called guano. And there is lots of guano found at bridges. These bats eat up to their own body weight and insects every single night. And those that are night roosting on bridges are using those features to digest their meal. So they tend to defecate before they go back feeding and leave behind lots of guano that we can sample. This is just another bridge. This is a very typical kind of bridge. So this is a concrete bridge, and up between these beams, there's these deep soffits that bats can roost in, and they trap warm air, and bats love to roost in these features. Sometimes we find large accumulations of guano right at the bridge ends. These are right at the bridge ends. There's these large blocks that the beams rest on, and they're called abutments. But they tend to accumulate large amounts of guano that just sits there. This is a This is an image of a bridge abutment where two beams come together, and where the two beams come together, that creates these narrow crevices where large numbers of bats were roosting. This bridge was in northern Alberta. And at this particular site, there was probably almost 5 gallons of guano across the bridge site, which is a huge accumulation. On concrete bridges, if you look on the underside, right up at the top corners, there's often guano stuck to these bridges that can be scraped off and used for diagnostic testing or DNA barcoding. They also roost on various other concrete surfaces, such as the diaphragms that connect the two beams. And this is the underside of the bridge deep in the soffits, and you can see guano stuck to the bridge beams there as well. Even steel bridges often still have concrete sections that the bats can grip hold of. They tend not to be found right on the metal, but they can find concrete at a lot of these sites anyways. These are very typical bridges found in the Northwest Territories. These ones were from somewhere in the Northwest Territories. There's not a whole lot of concrete that's easy to grip, except right at the bridge ends, there's a concrete back wall, and they can crawl behind the back wall of the bridge, and there's these concrete walls that they can grip a hold of and roost there. So as part of our project, we were collecting that guano. We can use a telescopic pole, so I don't have to get on the climbing gear and scale the side of a cliff. We can just use a telescopic pole to scrape the guano from the surface of the bridge. This guano stays stuck to the bridge probably for over a year. So what we're really sampling is a pretty wide cross-section of time. Which in some ways is perfect for detecting PD, because PD doesn't grow during the summer, it grows during the winter, but there's a decent chance that some of that guano will have been deposited early in the spring when they still have genetic material of a PD infection. So we think, or we have found, that this fungus can still be detected even when we're sampling late in the year during August or September or even October. This is just a close-up view of the sampling gear. We have different designs. The one on the right is a really narrow option for fitting between those box beams. Often the bats are not very high above our heads anyway, so we can just scrape it right off the sides of the beam. They tend to be found right at the bridge ends because those are the areas that tend to be warmest. And we collect that sample, and we have different objectives that we're using that guano for, and one of them is monitoring for the Pseudogenuascus fungus. But we can also use that guano for bat species ID. We can send it to a lab for DNA barcoding. The technology for DNA barcoding is getting better all the time, and we're at the stage now where we can submit a sample of about 200 fecal pellets, and they can return all of the bats, potentially return all the bats that contributed to that sample, even if it's just a single fecal pellet. And we can also use that sample for dietary analysis, and towards the end of this webinar, I'll briefly discuss that. But there's also a whole bunch of potential other objectives that we could address using that guano sample. The lab that does the DNA barcoding can also pull out rabies. They've developed a new assay for pulling out rabies virus within that sample. It could also be used for various ecotoxicology studies, and there's some projects in Alberta now using guano for examining pesticide exposure. For PD, we're aiming for about 25 milliliters of guano. It's actually quite difficult to find that amount of guano at a lot of sites, but the test can still be run with smaller amounts. So preliminary results across our study site, as of end of last year, we've surveyed over 770 bridges. Quite a few more have also been surveyed in British Columbia. So the total number is well over 800. And across Alberta, Saskatchewan, and Northwest Territories, we have about 436 bridges with at least some guano. You only need a single fecal pellet for DNA barcoding. Uh, so that is a huge sample size for examining bat species distributions. We also submitted quite a few of those samples for PD testing. So we tested about 119 bridges for PD, and we also submitted about 99 samples from other roost types, and those were typically buildings and bat houses. So far, we've detected PD at 9 sites, and 8 locations were inconclusive. So inconclusive PD detections are sites where PD was effectively found, but it didn't produce a strong enough signature to pass the diagnostic test. Basically, they had to amplify the DNA more than is allowed for it to be considered officially a confirmed detection. So this map shows where we found PD. The light red are the locations based on 2021 samples, and the darker red points are from the 2000 or 20 samples from 2022. And then the yellow samples are inconclusive samples from 2022, which we suspect are probably real detections based on the fact that they're occurring along the leading edge of where we expect PD to be spreading. One thing I want to point out, though, is we do not have very many samples from northern Saskatchewan or northern Alberta. So it could be right on the doorstep of Northwest Territories, and we simply would not know at this point. So it could be far more widespread in the north than we currently recognize. The uh, the pink line in the lower right-hand corner is the approximate distribution of this fungus based on our 2021 sampling. The upper line is based on our 2022 sampling, and it appears to have spread about 500 kilometers between the 2 years, which is quite a massive increase in its range. It's always possible that our sampling is inadequate to properly delineate its current distribution, but it's still a strong indication that it's spreading quite quickly across Western Canada. And if this rate continues, probably in the next 3 years, it could make its way up to the Northwest Territories. In Alberta, actually pretty much in any area, we expect a lot of the bat movements to occur along rivers. Rivers make these perfect travel corridors for bats. It's optimal bat habitat. There's lots of trees, there's water, there's lots of insects. So we expect one of the important travel corridors for bats to be along rivers. This is especially true in grassland areas where there's not a lot of tree cover. And sure enough, along the Red Deer River in Alberta, we find pretty consistent positive detections of this fungus. We have an inconclusive detection also along the Milk River, along the Battle River, along the Beaver River, and we've been tracking that, and we'll continue to track that this year to follow its spread. I'll quickly talk about the one confirmed detection we have in Alberta. This is along the Red Deer River, which have these really expansive eroded badland features, and these features create numerous cracks and crevices that are perfect for some bats to hibernate in. And stereotypically, we think of bats as hibernating in caves, but the reality is our largest hibernation sites may well be features such as the Badlands, where there's not large caves, but there's still lots of rock features that get below the frost line and in which they could hibernate. Yeah, so many bats in Western Canada, we think, are not hibernating in caves. We do know they hibernate in caves, but it's not necessarily the primary feature that they're using. This is the bridge that we detected PD at. This is near Dinosaur Provincial Park in Alberta. It's not a terribly remarkable bridge. Uh, they don't cling a hold of metal, but there's this concrete ledge that they were roosting along and deposited their guano. They also roosted in an open access port in the bridge itself. In case anyone is wondering, you can go right into that bridge beam. You could crawl to the other side, although you wouldn't get out again. But the bats are pretty maneuverable. They've gotten into this hole, and some of the bat guano was collected from within this beam itself. The last thing I want to talk about is the use of bat feces for DNA barcoding. Bridges are this really easy to sample location for getting bat guano, and the benefit of bridges is that they are on public land. So arranging access is really easy. You can basically just show up and collect it. As and what we were wanting to know is whether we could use samples collected at bridges as a way of monitoring or figuring out what arthropods occur in an area and potentially glean some information on what bats might be eating. So we're viewing this as a form of chirosurveillance or using bats to survey other groups. As so we submitted a number of samples across our study site. It was about 20 samples, but just 3 of those came from the Northwest Territories. But based on just 3 samples in the Northwest Territories, we were able to detect 11 orders of arthropods, 64 families, 125 genera, and 117 unique or species. Some uh, detections were only at the genus level, which is why there's more genera than there are species. But that's still quite a few species that we can detect from there. So for monitoring invasive insects, using bats can be a potentially powerful tool. Just as an example, based on just those 3 samples, we detected 6 different mosquito species in the region, and lots of moths. Moths are probably the favorite food of bats, although the bats that use bridges tend to be the myotis bats, which favor somewhat smaller moth species. So where do we go from here? We know that PD is spreading into Alberta and will probably spread into other regions fairly soon. And our priority right now is to monitor the impact that this fungus will have on bats. It's not guaranteed that the fungus will impact our bats the same way as has occurred in other regions. It probably will, but we can't assume that. And importantly, we need to figure out what species are being impacted. The endangered listing for little brown myotis and northern myotis was because those species occurred in eastern North America when this fungus first broke out. There are lots of other myotis species in western North America that potentially are just as impacted, and we need to figure out what impact PD will have on those species as well. And we there is some evidence based on research in Eastern North America that some bats appear to be resistant to this fungus, and there is hope that populations can stabilize and potentially recover. But for that to happen, we need to have the conditions in place that promotes high reproductive success and survival. So the priority of our program has been to encourage responsible bat management, ensuring that bats that use buildings can survive and are not excluded during improper times of the year, and to generally promote an appreciation of bats, because when people fear bats, they tend to implement poor management practices. That's all I have, and I look forward to any questions that people may have.
Thank you so much, Cory. That was an excellent, really interesting, if somewhat alarming presentation. I personally was definitely concerned to hear just you said that white nose or the fungus could potentially reach the NWT within 3 years, which is not a lot of time. So I'll open things up. If folks on the line have questions, please put them in the Q&A box, and I will take a look at those for Cory to answer. In the meantime, I'm not seeing any yet, so I'll just I'll ask one, which given this timeline and the ongoing spread of PD, what are some things that we can do, both like as researchers, as you said, the original introduction of PD could have been from a researcher who got it on their gear and moved it around. So what are some things that we can do as researchers to protect bats, as well as just the general public? You touched on that a bit, but if you could go over some kind of best practices.
Yeah, so proper decontamination is important, and I think it was around 2016 or so. I could have the year wrong, but the fungus spread all the way from Eastern North America to Western North America and started spreading from around Washington State. And we think that was probably caused by people. Maybe it was someone visiting a cave and then spreading it, or it could have just been a bat hitching a ride in a long haul vehicle. But those sorts of long distance translocations of this fungal material needs to be avoided. So if you enter a bad habitat in one region, you need to thoroughly decontaminate before moving to another region. The Canadian Wildlife Health Cooperative has decontamination protocols and recommendations for how to do that. So that's important. Now, having said that, most of the spread is currently from bat to bat, and the fungus is going to spread regardless of what people do. It's not, like I said, it's not guaranteed that it will become widespread in northern climates. It's quite possible, though, that it will, and we'll need to monitor and see what is happening there. So this is, in some ways, this is a little bit unfortunate because a lot of the management for trying to get rid of this fungus is beyond our control. There are some projects going on to explore the potential use of a probiotic treatment. WCS Canada has a project going on in British Columbia and Washington State to see if probiotic treatments could help give bats some resistance. And basically what a probiotic is you collect beneficial microbes from the native habitat and try to inoculate bats in hopes that those beneficial microbes could give some resistance to this fungus. But that's a pretty speculative treatment at this point. It hasn't been demonstrated to be effective. So controlling spread is about the best we can do currently.
Mm-hmm. Right. Well, thank you. I've got some questions in the Q&A now. Joanna Wilson, who we both know, has said, great, thanks for the great presentation, Cory. 2 questions. And Joanna, I only see one of your questions, so feel free to submit the other one if you like. But the first question, potential use of guano for rabies surveillance is interesting. Were the NWT samples submitted for this testing, or could we do that in the future?
I would think they could be submitted for testing. I would have to have a discussion with the lab to make sure that it's ready for deployment to new regions. I think they were just working on this test. So if that's something that is of interest, I can certainly look into that. There's a number of other things that would be interesting to look for, and the other one is histoplasma, the fungus that causes histoplasmosis in people. And it's not certain that even if it's present in a region that it would be present in samples from bridges, but these are potential other applications that could be looked at in the future.
Interesting. Something more to follow up on, maybe, Joanna. Okay, the second part of Joanna's question, how likely do you think it is that inconclusive PD results could be false positives or a similar fungus species as opposed to weak PD signals?
Yeah, I don't have an answer to that. I'm not heavily involved in the actual testing of the sample. Now there are fairly rigid protocols that the lab follows based on practices done elsewhere. And I know I was told in British Columbia they did bind signatures of non-target Pseudogymnoascus species. Uh, the sequence they're using for identifying this fungus should be fairly unique to the species, but it's not always the case. Sometimes different species do share the same barcode. Um, but yeah, hopefully that's not the case. Now, in our case, the positive PD detections are following a pretty predictable pattern, spreading from Manitoba, and then it kind of progresses a little bit west between the 2 years. I think if it was detecting a non-target uh, uh, Pseudogymnoascus strain, I think we would see it randomly popping up in locations that don't make any sense.
Right. Yeah, that makes sense. Another question for you. Do you feel the environmental conditions in Hibernacula in the NWT may be significantly different enough from Alberta or other southern sites that PD or white nose syndrome may have less of a chance of getting established in the NWT?
Yeah, the short answer to that is that I really don't know. As you go north, the average hibernation or the average cave temperature should go down. But there is a little bit of heterogeneity as you move across the landscape, and it could be that they're still selecting warm caves where this fungus would grow quite well. I was trying to find some temperature data on some of the caves around Wood Buffalo, and I couldn't track it down, although I'm sure it exists, just to figure out what the average temperature there was. But I think there is a good chance that it could differ. So what I think is important is that we don't just assume that it's going to have the same impact, and we implement monitoring to figure out what that impact will be.
Right. Okay. I don't see any more questions. I'll give folks a little more time to. Okay, there's a question in the chat, so I will read that one out. Um, can we treat bats in caves with ultraviolet light to kill the fungus?
I don't believe that has any practical application currently. It would not be easy in large part because when they're in caves, they're often deep in rock crevices. So most of the cave you wouldn't be able to actually reach or hit with the UV radiation anyways. But even more important than that, we don't know where most of the caves are. If you were to add up all the known hibernation sites for bats across Western Canada, I don't even think it would reach 10,000 bats. And we probably have millions of bats. So uh, we effectively don't know where most of our bats are. So any targeted treatment to a cave is probably going to be a drop in the bucket.
Right. Okay. Yeah, if folks have any more questions, please put them in the Q&A. Those are some great questions we've had so far. I have another one that I will ask to give folks another minute or two if they want to put any last questions in the Q&A. You mentioned near the beginning of your presentation that bats in Europe don't get white nose syndrome. I was just wondering if you could kind of elaborate on that a little bit. Are there any implications for, you know, does that mean anything about how bats in North America might be able to become resilient to this disease?
Mhm. Yeah, so the fungus PD is quite widespread in Europe, but they don't get the disease. And I believe the current hypothesis is that they probably had this fungus at some point in the distant past, and they developed resistance to that fungus for it might be a behavioral change, or maybe it's some physiological change, or small scale evolution. But yeah, they're not susceptible to white nose syndrome. And probably if we wait another few hundred years, the same will happen in North America. The question is whether they can recover their populations in light of all the other pressures that they're facing as well. So there's issues with declining insect populations, habitat loss and degradation, human persecution. So those additional pressures need to be managed to give them the best chance of recovery.
Right. Yeah, I think overall the takeaway is that we need to be proactive and take care of our bats. And I think everyone on this webinar is probably all for that, but more outreach is always a good idea, and spreading the word that we love bats and they are great. And we would like to keep them healthy in the NWT and everywhere.
Mm.
Okay, well, I'm not seeing any other questions in the Q&A or the chat, so I think we can wrap up here. Thank you again so much, Cory, for that excellent presentation. And also thank you to everyone online who joined us today. If you'd like to join us for more great webinars happening this week, you can find more information on our website, nwtkisp.ca. That's nwtcispp.ca. Or you can look us up on Facebook. Our Facebook page is NWT Council on Invasive Species, Pests, and Pathogens. With that, thanks again, Cory, so much, and everyone have a good day.
Yeah, thank you.
All right, thanks.